Hello friends, this is principle of colorimetry, as well as we will be seeing hands-on training on how to measure a colored solution, the concentration of a colored solution using colorimeter. Now this particular instrument, the colorimeter, was first introduced by Jules Dubosc in 1870, and colorimeters are used to detect color and hence concentration of solutions, colored solutions, using a monochromatic light. Now, monochromatic light means light of a single wavelength. Now, when a monochromatic light is passed through a colored solution, the colored substances or molecules in the solution will absorb a part of that light, and a part of that original light will be transmitted or emitted also. Now, in biochemical measurements, the substances are detected, or concentration is detected basically by color development. So, some sort of color is developed using some chemicals for glucose or carbohydrates, other carbohydrates or protein or amino acids. Some color is developed, and the intensity of that color is red or detected. And this is colorimetric measurement is based on two principles: Beer's law and Lambert's law. Beer's law. Now, Beer's law states that when a monochromatic light is passed through a solution, the intensity of the emitted light is decreased exponential to the concentration of the substance present in the solution. Which means if the number of molecules, absorbing molecules are higher, the color is higher, the absorbance also will be higher. Lambert's law states that when a monochromatic light is passed through a solution, the intensity of emitted light decreased exponential to the thickness or path length of the cuvette used. This means that when the liquid column, usually the path length of a cuvette is 1 centimeter. If the liquid column or the path length is higher, the absorbance also will be higher. Parts of colorimeter. This light source, the most common source of light used in a colorimeter is a tungsten filament. Monochromator, glass filters are used to select the particular single wavelength or monochromatic light. Sample holder is a cuvette made up of glass are used to hold the colored solutions. Cuvette with a sample is placed in reading chamber of colorimeter for reading. Photodetector system or photoresistor. When emitted light falls on the detection system photoresistor, an electric current is generated. This is exhibited in the galvanometer as reading. Now, measuring device, the galvanometer shows the meter reading that is directly proportional to the intensity of light. A microprocessor then calculates the absorbance or percentage transmittance. Now, this is a chart showing color of the solution and the complementary filter to be selected. If the color of solution is blue, you have to select yellow or red color. Likewise, green solution, red filter, yellow solution, blue filter, red solution, green filter, and if the solution is purple colored, you have to select yellow colored filter. Calculation. The absorbance or optical density is taken as the index of measuring concentration of samples. Absorbance is not having a true unit, and you can see the formula there. OD of test divided by OD of standard multiplied by amount or concentration of standard used for the test divided by volume of test used in the experiment. Then the result also you can see there. It is expressed in milligrams per ml, which can be multiplied by 100 to give milligrams per 100 ml. Now you can see the one of the examples also there. 0.31 as OD of test, 0.41 as OD of standard multiplied by 0.5 milligram per ml standard concentration divided by 1 ml test used. The result is 0.37 milligrams per ml, which will be multiplied by 100 to give 37 milligrams per 100 ml of concentration. But the most accurate method of measuring the concentration of an unknown solution is by preparing a standard curve graph from a series of standards of different concentrations from a stock standard. This is much more accurate than calculating from OD of a single standard. Optical density of each standard and the test sample are taken, and a curve is plotted taking OD on the y-axis and concentration on the x-axis. The unknown concentration of the test can be obtained from the curve using its OD, that is red. So that makes two types of calculation of results. You can go through this chart that shows the difference between colorimeter and spectrophotometer. It's a question that is usually asked for theory and practical exams. Now we can see how a colorimeter practically works. Now basically, we do some color reactions in biochemistry to detect substances. And you can see here a series of test tubes having a gradient of colors is placed. We're actually seeing a series of standards taken for the purpose of measuring using colorimeter. Our target here is to measure the absorbance or optical density of these colored solutions, which will be done with a specific wavelength filters and a colorimeter. And now you can see a typical brand colorimeter switched on to warm up for about 5 minutes before calibrating. This is colorimeter. Keep in mind that never pronounce the instrument name as calorimeter. It's a different instrument used to measure specific heat in physics. Now, here you can see a series of standards and test solutions. This is for the purpose of demonstration using colored solutions. You can see here the difference in color of these solutions are observable, and it's not so observable here. So this is when the instrument actually comes into play to differentiate the exact absorbances. This is a cuvette made of glass of uniform material, and it will be having a marking on it, which should coincide with the marking on the cuvette holder of colorimeter. And before measurement, an appropriate filter is to be chosen from filter disc of colorimeter. Typical range of wavelength used in standard colorimeter is 420 to 710 nanometers, ranging from blue to red in visible light. That is 420 around is violet blue, and red is around 710 nanometers. And this is the marking on the cuvette that should coincide with the colorimeter marking, so that there will be a regularity in passage of light while we read colored solutions. And using the operations button, we can choose mode, which is kept in OD, as you can see the blinking of light there. Another important button is that of auto zeroing, and we can see the display unit also there. Now we can proceed with the zero setting using blank solution. 1 ml of blank is transferred to a dry, clean cuvette, and this is to be read and made zero by pressing the press button on the colorimeter. Now blanking is done to cancel out the OD contributed by other reagents involved in the experiments. So blank is also called a reference solution, so it avoids the interference of other reagents. Now the zero is set now. Now here, make it a practice that after taking reading of a solution, you pour it back to the same tube, tap the cuvette on the tissue paper, and rinse the cuvette with the next solution that is to be read in the instrument. So like this. You can discard that solution. As also, you have to wipe the cuvette. After adding the next solution, you have to wipe the cuvette with a tissue paper before next reading to get rid of water or fingerprints which may be there on the cuvette, glass cuvette. Now on, we can take the OD of standards and all the colored solutions, like placed in the cuvette holder. The OD comes at 0.08. Another colored solution, a standard or test, whatever, reading 0.18. Another solution, the reading is 0.42, like that. A blank is a solution having all the reagents in it except the test substance. And a blanking can be done in between reading also, because if there is a drift in zero setting, that can be adjusted. As well, if you use a new filter, you'll have to blank again. Now, during reading of colored solutions, if the absorbance goes above range, over range, you'll have to dilute the solution and read it. So this will enhance the accuracy of results. So that's all about the principle and operation of colorimeters. Thank you for watching my video. Thank you very much.